In this video class, we will discuss about different forms of DNA. Our previous class was about the double helical form of DNA proposed by Nobel laureates Watson and Crick. I hope all of you have watched that, and if you have any doubts regarding that, please ask. Okay. Then I have said in the previous class this typical or classical type of double helical model is known as B form of DNA or BDNA, and it is the first form of DNA today we are going to discuss. This is the typical or classical type Watson and Crick double helical model. It is also known as B form of DNA or BDNA. And you all know that BDNA is right-handed double helix. And what is the diameter of the double helix? It is 20 angstrom. Then length of one complete turn. That is, it's also known as pitch of helix. It is 34 angstrom. Please note that in some textbooks, instead of this angstrom, they are given the length in nanometer. Because 34 angstrom is equal to how much nanometer? How many nanometers? 34 angstrom is equal to 3.4 nanometer. Okay, in the same way, the distance between two consecutive base pairs, 3.4 angstrom, it is equal to 0.34 nanometer. So don't get confused when you see nanometer in place of angstrom in any textbooks. Both are same. Okay. Then base pairs per turn, it is 10.5. And distance between two consecutive base pairs, that is also known as axial rise of base pair, it is 3.4 angstrom. And tilt of base pair, it is 6.3. And we have discussed about two grooves in the double helical structure. One is major groove and other is minor groove. And the width of major groove, it is 12 angstrom and depth is 8.5 angstrom. And minor groove width is 6 angstrom and depth is 7.5 angstrom. See this. This is the minor groove and this is the major groove. Okay. And length of one complete turn or helical pitch, it is 34 angstrom. What do you mean by one helical turn? It's rotational 360 degree. So it is 34 angstrom. What is the diameter of the helix? It is 20 angstrom. See here, it is a minor groove and a major groove. And the distance between two consecutive base pairs or the axial rise, it is 3.4 angstrom. And base pair tilt is 6.3. And there are approximately 10.5 base pairs per turn. That's all about the typical or classical form of DNA double helix, BDNA, right-handed DNA double helix or BDNA. Then we will pass on to other alternative conformations of DNA. The X-ray analysis of DNA crystals at atomic resolution have revealed that DNA exhibits much more structural diversity. And such variations are ADNA, ZDNA, CDNA and DDNA. And of this, ADNA, CDNA and DDNA are right-handed double helix, just like our typical model BDNA. ADNA, CDNA and DDNA are right-handed, whereas ZDNA, it is left-handed double helix. We will discuss one by one in detail. First comes ADNA. It is a right-handed double helix. And see this figure. What is the difference between ADNA and BDNA? See this. What is the phenotypic difference? This ADNA, it is much broader and more compressed along its axis than BDNA. You can clearly see here this ADNA, it is much broader and it is more compressed than BDNA. More compressed in the sense that ADNA is 20 to 25% shorter than BDNA. And ADNA, it occurs only in the dehydrated sample of DNA, such as though used in crystallographic experiment. And coming to the grooves are not much deep as in BDNA. There are two grooves, one is major groove and the other is minor groove, but the grooves are not as deep as in BDNA. Here, see the difference. Here the grooves are much deeper, but in this ADNA, it is not that much deep. A major groove is deep and narrow and minor groove is wide and shallow. Then, what is the diameter of the double helix? It is 23 angstrom. For BDNA, it was 20 angstrom. So, for ADNA, it is 23 angstrom. And length of one complete turn or pitch of helix, it is 28.15 angstrom. And base pairs per turn, it is 11. And for BDNA, it was 10.5. And distance between two consecutive base pairs or the axial rise of base pair, it is 2.45 angstrom. For BDNA, it is 3.4 angstrom. And tilt of base pair is, it is 20.2. See the table, see the comparison table. BDNA, this is the typical form of DNA or the Watson and Crick, the double helical form of DNA. Okay, so it is right-handed, twist of strands. This is right-handed, a clockwise twist. ADN is also right-handed, CDN is also right-handed, BDN is also right-handed. Then what is the diameter of the double helix? BDNA, for BDNA it is 20 angstrom and for ADN it is 23 angstrom. For CDN it is 19 angstrom. And about the pitch of the helix, that is length of one complete turn. For BDNA it is 34 angstrom, ADNA 28.15 angstrom and CDNA it is 31 angstrom. This D type of DNA, it is very extremely rare variant. Okay, so most of the information is not available. So base pairs per turn. For BDNA it is 10.5 base pairs per turn, ADNA 11 base pairs per turn, and for CDNA it is 28 by 3 base pairs per turn, and for DDNA there are only 8 base pairs per helical turn, and DDNA, D form of DNA shows 8-fold symmetry. Since there are only 8 base pairs per helical turn, you can say D form shows 8-fold symmetry. Then axial rise of the base pair, that is distance between two consecutive base pairs. For BDNA it is 3.4 angstrom and for ADNA it is 2.5 angstrom, CDNA 3.32 angstrom and for DDNA it is 3.03 angstrom. And coming to the tilt of the base pair, BDNA it is 6.3, sorry, no angstrom here. Okay, this is by mistake, 6.3. This is not here. Okay, omit this. Okay, then this CDNA and DDNA show a negative tilt. And what do you mean by negative tilt? In the negative tilt, the base pairs are displaced backwardly with respect to the axis of the DNA double helix. So for the CDNA and DDNA, they show negative tilt. CDNA 7.8 and for DDNA it is 16.7. The A form of DNA is found at 75% relative humidity in the presence of sodium, potassium and ions. The C form of DNA is found at 66% relative humidity in the presence of lithium and magnesium ions. And we have already said about DDNA. DDNA is an extremely rare variant with only 8 base pairs per helical turn. So D form shows 8-fold symmetry. Then coming to ZDNA, the most important type of DNA, ZDNA, because all other alternative conformations which we have studied, they are right-handed. We have studied BDNA, the typical form, then ADNA, CDNA, and BDNA. All these four types are right-handed double helixes. But when we come to the ZDNA is left-handed zigzag double helix. Z for zigzag. Why it is known as ZDNA? Because the sugar phosphate backbone presents a zigzag conformation. See here is a zigzag conformation. You can clearly see a zigzag conformation, isn't it? So ZDNA shows the sugar phosphate backbone shows a zigzag conformation. So it is known as ZDNA or zigzag DNA. And it was discovered by Andrew Wang, Alexander Rich, and their co-workers in the year 1979. Then, just like BDNA, the strands of ZDNA are also anti-parallel. And this see this picture. What's the difference between BDNA and ZDNA? Compared to BDNA, ZDNA it is long and thin. Compared to BDNA, ZDNA is long and thin. And how many grooves you can see here? Only a single groove. Major groove, either the major groove may be rudimentary or it is completely absent. So we can say major groove is absent. Only minor groove is present in ZDNA, and that is the major difference between BDNA and ZDNA. That is one of the major difference between BDNA and ZDNA. In BDNA, there are two grooves. One is minor groove and the other is major groove. But in the case of ZDNA, only a single groove is present and it is minor groove, and major groove is absent. Then the non-rotating configuration of the base is called anti-configuration and the rotating configuration is called syn-configuration. So in the case of BDNA, the configuration is anti-configuration, and for ZDNA, it is syn-configuration. And what is the diameter of the double helix for ZDNA? It is 18 angstrom. Then length of one complete turn or pitch of helix, it is 45 angstrom, and base pairs per turn is 12. For BDNA, it was 10.5. And distance between two consecutive base pairs or axial rise of the base pair, it is 3.7 angstrom. Just compare this with BDNA. What is the diameter of the double helix for BDNA? Okay, the diameter of the double helix for ZDNA is 18 angstrom, and what is the diameter of the double helix for BDNA? It was 20 angstrom. Then length of one complete turn. For BDNA, it was 34 angstrom, and for ZDNA, it is 45 angstrom. And base pairs per turn in BDNA is 10.5 base pairs per turn, and for ZDNA, it is 12 base pairs per turn, and distance between two consecutive base pairs in BDNA is 3.4 angstrom, and for ZDNA it is 3.7 angstrom. When you study ZDNA, always study by comparing with BDNA, because BDNA is the typical or classical type, and BDNA is right-handed and ZDNA is left-handed. So always study these two by comparing. What is the difference between BDNA and ZDNA? Both are very, very important. BDNA is important, ADNA is important, then ZDNA is also very, very important in the exam point of view. Okay. This D form and C form are very rarely asked. Sometimes they will ask the differences between B, A, C and D. But it is very rarely asked. Mostly questions come from from this part, from BDNA, ADNA and ZDNA. So don't get confused. You study by comparing these two, ADNA and ZDNA with BDNA. Mostly focus on BDNA and ZDNA, because BDNA is right-handed and ZDNA is left-handed. Then another important unique feature of ZDNA is that it is composed of repeated guanine cytosine sequences on one strand with complementary cytosine guanine sequences on the companion strand. And this is one of the unique feature of ZDNA. And coming to the functions of ZDNA, it is involved in the genetic recombination of eukaryotes. The stable form of ZDNA is involved in genetic recombination. How ZDNA is stabilized? It can be stabilized through methylation of its cytosine residues. Okay, so first function, it is involved in genetic recombination in eukaryotes. It also serves as recognition signals during DNA replication and transcription. So the functions include genetic recombination and also recognition signals during DNA replication and transcription. Then till now we have discussed about the double helical structures of DNA, the typical BDNA, then ADNA, ZDNA, CDNA, BDNA. All these are double helical structures. And now we are going to discuss the triple helical model of DNA. DNA or the DNA triplets. Okay, see this figure. This is a triplex DNA structure. The first strand, second strand, and third strand. So, come to the point. The triple-stranded DNA, also known as HDNA or triplex DNA, is a DNA structure in which three oligonucleotides wind around each other and form a triple helix. And triple-stranded DNA is also known as HDNA and H for hinged DNA. And it is the unusual type found in the genomes of vertebrates and invertebrates. And here, in the triple-stranded DNA, the third strand, these two green colors, this is double-stranded DNA, and this violet color, it is the third strand, isn't it? And this third strand binds to the B form of DNA by forming Hooke's-Steen base pairs or reversed Hooke's-Steen hydrogen bonds. We will discuss what is Hooke's-Steen base pairs. Just remember this term. It's very important. Okay, and see this triplex DNA structure. The arrows are going from 5' end to the 3' end. So 3', 5', 5', 3', 3', 5'. This is the third strand which binds to the B form of DNA. Then we will discuss now what is Hooke's-Steel base pair. Here you see this picture. What is this? This is adenine and this is thymine. Isn't it? This is the base pairing between adenine and thymine. And what happens in triplex DNA is that a thymine nucleobase can bind to a Watson and Crick base pairing of thymine adenine by forming a Hooke's-Steel hydrogen bond. So this is Hooke's-Steel hydrogen bond. Okay, this is the normal base pairing between adenine and thymine in the double helical model of DNA. And another thymine comes and bind to this adenine and thymine base pairing by forming Hooke's-Steel hydrogen bond. And the thymine hydrogen bonds with the adenosine of the original double-stranded DNA to create a PAT base triplex. So please note that in triplex DNA there are 3 strands. Of this, 2 strands contain pyrimidine and the third one contain purines, or vice versa. That means it may contain, the 2 strands of it contain purine and 1 strand pyrimidine. 2 pyrimidine, 1 purine, or 2 purines, 1 pyrimidine. Okay. Adenine is purine and thymine is pyrimidine. So 1 purine, 2 pyrimidines. The formation of HDNA is stabilized under acidic conditions and the presence of divalent cations such as Mg2+. So in the triplex form or the HDNA or the hinged DNA, an extra nucleobase comes and binds to the Watson and Crick normal base pairing by forming a Hooke's-Steel hydrogen bond. Remember this name, Hooke's-Steel base pairing. So in triplex DNA, what is the base pairing? It is Hooke's-Steel base pairing. Functions of, coming to the functions of HDNA, DNA triplex, it is commonly found near the sequences involved in gene regulation. So it plays a role in RNA synthesis, initiation and termination of replication and recombination. These are the functions of the DNA triplex. Then now we have studied double helical model, triple helix. Then next we are going to study DNA tetraplex or quadruplex. Okay, so what do you mean by DNA tetraplex? DNA tetraplex, they contain the sequences which are either guanine rich or cytosine rich. Okay, DNA tetraplexes, they contain sequences which are either guanine rich or cytosine rich, and they are found at the telomeric ends of chromosomes. And these sequences are of fundamental importance in protecting the cell from recombination and degradation. So we will discuss one DNA tetraplex, that is I-motif DNA. It is a tetraplex or quadruplex. It is a 4-stranded DNA structure. Okay, so this is important, I-motif DNA. And what does I indicate? I means intercalated motif DNA. And this I-motif DNA, it contains cytosine-rich DNA sequences, and the cytosine-rich DNA sequences can fold into a structure called I-motif. See this here, this is cytosine-rich DNA sequences, and this can fold into a structure called I-motif. And that structure, cytosine rich structure, it is four-stranded quadruplex DNA structures, similar to guanine quadruplex structures that are formed in guanine rich regions of DNA. Okay, and these structures typically appear in promoter regions on the DNA. These structures, these type cytosine-rich DNA sequences or I motifs, they are seen to appear in promoter regions on DNA. And what is the difference between the base pairing in DNA double helix and this I motif? In this four-stranded DNA structure, the cytosine on the same strand of DNA bind to each other. It will not bind to guanine. Okay, cytosine on the same strand of DNA bind to each other. Cytosine bind with cytosine itself. Cytosine will not bind with guanine. But what happens in DNA double helix? In DNA double helix, uh, cytosine binds with guanine in the opposite strand. Okay, the letters on the opposite strands, they recognize each other and cytosine binds to guanine. But in this I motif structure or the quadruplex DNA structure, the cytosine pairs with itself by forming three hydrogen bonds. And this coming to the structure of I motif, it sequences CCCCC. Okay, and the two duplexes combine in an anti-parallel manner. The combination occurs by intercalation or insertion of cytosine-cytosine base pairs. This we have already discussed. Cytosine-cytosine base pairs. And cytosine-cytosine base pairs are bonded by three hydrogen bonds and which form stronger bonds than conventional GC pairs. So, the bond formed in between cytosine-cytosine is more stronger than the bond which is formed between cytosine and guanine. The base pair energy of cytosine-cytosine bond in an I motif is 169.7 kilojoules per mole, while the base pair energy of Watson and Crick GC model, the GC base pair, it is 96.6. And the structure of I motif was derived by using nuclear magnetic resonance. The length of cytosine sequences ranged from 3 to 12 bases. It may also have timing residues in between the cytosine sequences. So the length of cytosine sequences range from 3 to 12 bases. Timing residues are also seen in between the cytosine residues. See timing here. Okay, cytosine timing. Then what are the functions of I motif? They play a major role in gene regulation and expression, telomerase inhibition, DNA replication, and repair. So, we have discussed double-stranded DNA, DNA triplex, DNA tetraplex. What is the example for DNA tetraplex? It is I motifs. And what is the example for DNA triplex? It is HDNA. Then Watson and Crick's double helical model of DNA. The typical form is BDNA. BDNA is right-handed, whereas ZDNA is left-handed. BDNA, ADNA, CDNA, and DDNA are right-handed, whereas ZDNA is left-handed. Then, you know, single-stranded DNA is found in certain viruses, such as bacteriophage, which consists of single-stranded circular DNA. Okay, it becomes double-stranded only at the time of replication. This we have already discussed in our previous video class when we are discussing about the Watson and Crick double helical model. and just go through this table. This some basic differences between double-stranded DNA and single-stranded DNA. Just go through this. This is only for your understanding. That is, double-stranded DNA it remains in linear helical form, whereas single-stranded DNA remains in circular form. However, it becomes double-stranded only during replication. That is, replicative form. We will discuss this in detail during when we study DNA replication. Okay, and one important difference is that the base pair composition in double-stranded DNA is equal. That is, uh, purines are equal to pyrimidines and pyrimidines are equal to purines. Adenine is equal to thymine and guanine is equal to cytosine. But in single-stranded DNA, these are not equal. The composition of adenine, thymine, guanine and cytosine is in the proportion 1. 1 for adenine, then 1.33 for thymine. For guanine it is 0.98 and for cytosine it is 0.75. Okay, so these are the some basic differences between double-stranded DNA and single-stranded DNA. So that is all about today's class. So in this class, we have discussed about different forms of DNA, and in that the typical or classical model, B form of DNA is important, A form is important, and the left-handed model, that is ZDNA, is also important. Then DNA triplex, remember DNA triplex HDNA and DNA tetraplex I motifs. Okay, so I hope you have understood the class, and if you have any doubts, please ask. And in the next class, we'll meet with another topic. Till then, bye bye. Have a nice day. Thank you.